Good morning students. So, welcome to the next lecture of biomolecular spectroscopy. In the last lecture we studied how we can use circular dichromacy spectroscopy to understand one of the intermediate state in protein folding, which is called molten globular state, and how we can combine with other techniques such as fluorescence, ANS fluorescence, DLS and all those to understand the molten globular state. So protein folding is very interesting phenomena, and protein misfolding can lead to many of the neurodegeneration diseases, right. So, brain disorder, and some of those brain disorders can come from the protein structure called amyloids. Amyloids means amyloid term came because of they were like a starch like property, that is what amylene, amylase and all those that, and so it was infectious starch like looking particle, proteinaceous particle that was found in many of the brain, which degenerated because of the presence of this protein deposit, and this is the one of the conformation that is responsible for toxicity in neurons. So, the protein basically aggregates and forms amyloid like fiber, and that is in neurodegeneration disease. So, how we can use circular dichroism in neurodegenerative disease, that is what we are going to discuss today, right. So, neurodegenerative disease, just to refresh your memory, it is a group of incurable, at the moment it is incurable, right. So, there is no certain medication that has come up which can cure neurodegeneration. So, it is a group of incurable disease, progressive condition characterized by the gradual loss of structure and function of neuron in the brain and peripheral nervous system. So, if neurons are damaged, everything gets into to halt. So, all the behavior, all the action and all those. So, these disorders, so neurodegenerative disorders leads to decline in cognitive, that means understanding. So, dementia this is called, motor, that is a movement capability, this happens in Alzheimer's disease, this happens in Parkinson's disease, and it often gets worsened with time. So, with age it goes into severity, and then it becomes a very chronic conditions. So, some of the example of this neurodegeneration: Alzheimer's where memory loss happens, Parkinson's where motor movement get damaged, and ALS. So, AD, it is the most common form of dementia, primarily affecting the memory and cognitive function. PD, Parkinson's disease, characterized by tremor, rigidity, slowness in movement, movement related problem. ALS, it is a motor neuron disease causing muscle weakening and atrophy. And the Huntington disease, it is a genetic disorder leading to uncontrollable movement and cognitive decline. Then multiple sclerosis, it affects central nervous system, causing motor and sensory issues. So, most of these are neurodegenerative disease, and in each of these disease one or other protein gets aggregated, forms amyloids, and that is the root cause for neurodegeneration. Right. So, say for Alzheimer's disease, there are two proteins, amyloid beta and tau, that segregate. In Parkinson's disease, the main culprit is a protein called alpha synuclein. In Huntington disease, it is a protein called huntingtin. In ALS, it is a TDP43. So, these proteins actually forms a toxic aggregate, and that disrupts the neuronal function and finally leading to the cell death. That is how progressive damage in the neurons start happening, right. So, what happens that amyloid beta, which is in Alzheimer's disease, forms an extracellular plaque in the Alzheimer's disease. The tau, again it is a 441 long amino acid long intrinsically disordered protein, forms the neurofibrillar tangle inside the neurons in Alzheimer's patient and other tauopathy that are related to tau. The synuclein, it is a 140 amino acid long protein, it accumulates in the aggregated form as a Lewy body in Parkinson's disease and dementia related with Lewy body. The huntingtin HTT, it is mutated with extended polyglutamine. So, Q, many Q comes repeat and that gets segregated in Huntington disease. The TDP4, it is a TAR DNA binding protein 43, TDP43. It accumulate in amyotrophic lateral sclerosis and frontotemporal lobe dementia happens. So, in case of the ALS. Then prion protein, it is a very nice folded protein, but when it unfolds, one of the helix converts into sheet and then it starts aggregating, and this misfolded form cause a disease called Creutzfeldt-Jakob disease or other prion disease. The FUS protein fused in sarcoma, this is associated with some form of ALS. Then there are other markers like elevated blood level of GPFF, NEFL, GDF15 and so on and so forth. So, the common generic feature, it appears that many of these proteins which were otherwise intrinsically disorder nature undergoes into major conformational change. From intrinsically disorder state, they go to a cross beta structure, creating this beta sheet rich aggregates, and that gets deposited in the brain, and they can also propagate like prion protein from one cell to another cell, and that is what happens in neurodegeneration, right. So, how we can study these amyloid protein using CD. So, now CD it is a very simple tool that we have studied in last 4-5 lectures. It is a critical tool for understanding the structure, aggregation kinetics, inhibition of amyloid fiber, what is the potential molecule that can inhibit the amyloid formation. So, those we can study in context of neurodegeneration like AD, and all those, and how the movement or transition of this intrinsically disordered protein, that is random coil monomer, to toxic beta sheet rich amyloid fibers happens. So, the key aspects in CD amyloid research is, can we monitor the structural change that is happening from disordered state to the beta sheet rich amyloid fibers, right. So, CD can track that transformation of a protein, say A beta, which is in Alzheimer's disease, and what we have to do, just look at the random coil band, how it is changing to beta sheet structure at 217 to 18. You can also do the inhibitor screening, right. You can employ how we are breaking the amyloid fibers or arresting that protein into the disorder state. So, suppose cyclodextrin liquid ionic crystal can inhibit the formation of these toxic polymers. So, we can incubate the protein and look at their structure. Then we can do time resolved analysis, how we can use the CD to monitor over the time the protein which is aggregating, what is the lag phase, log nucleation phase, and then maturation phase, one can do that. And then you can even refine the technique. It is limited in large aggregates, right, scattering starts to happen. So, in those case, how we can use some of those algorithm like we discussed Bestel or CDNN by analyzing those as a thin film to get a high accurate, high quality data. So, let us take one of the example of amyloid beta protein which is present in the Alzheimer's disease, right. So, amyloid beta basically it is there are various variant A beta 40 or A beta 42. So, let us take a simpler fraction, say 36 to 43 amino acid long, one of these, and these are derived. So, the various fraction that we discussed, A beta 40, A beta 42, they are derived from a protein called amyloid precursor protein APP that plays a central role in AD. So, alternate splicing that happens by, or the chopping up happen by two protease called alpha beta secretase that cuts the APP and releases this 36 to 43 amino acid long amyloid beta protein. So, this accumulates in the brain, forms the toxic insoluble extracellular plaque that disrupts neuron function, and that is a hallmark of Alzheimer's disease. CD can do that transition or structural change that happens. What happens that we want to probe how a intrinsically disordered protein goes to fibril. So, what we can do? We can do the real time monitoring of conformational change, how like a random coil structure goes into beta sheet structure. Then we can understand the detailed kinetics, which fractions say A beta 40 or A beta 40 through 2 or something like that, which is aggregating faster. So, like we can monitor the structural transition that goes from say random coil state to a beta sheet state, right. So, that is what one can do it, and if we know that some of these say familial mutants, that is cause of the early onset of disease, how those could be, because their aggregation kinetics could be different. So, one can basically using CD we can evaluate even the inhibitor that does not allow this conversion from random coil to beta sheet. So, what kind of different compound, small molecules, ionic liquid, drug that can inhibit the A beta aggregation, and what structural transition that happens because of the presence of that. So, advantage of CD in a study of amyloid beta, you can like a very less sample is required, and you can do a quantitative analysis, like you can use the advanced analysis software like Bestel. You can combine Bestel with other deconvolution technique to quantitatively evaluate the secondary structure structure that can change in A beta, and you can distinguish once it goes from random coil state to the cross beta structure, what kind of structure it is populating, whether they are populating parallel structure or anti-parallel structure, what kind of structural transition happens, right. And you can use this in various aggregation kinetics, like looking at the stability, aggregation of different amyloid related protein and so on, right. So, and like as I was saying, you can use this sample types with various buffer and all those, and look at solid phase if the aggregates are formed. So, one can do that. So, let us take some of the example and evaluate the performance of ionic liquid as an inhibitor of amyloid fibril, as a stabilizer of beta amyloids using CD spectroscopy and other program. So, this is our A beta, right. So, suppose we purchase this from a company, this is the 40 amino acids got synthesized. If you look at, it is a mostly disordered protein, that is what we are getting the A beta form of amyloid. Then there are some ionic liquids that are there which forms the ionic liquid, and these are some of the commonly used ionic liquid that suppose we have, right. And now we want to probe how this protein in absence and in presence of ionic liquid goes in aggregation mode. So, a structural transition takes place. So, we are using say a CD from JASCO J1500 with a Peltier which controls the temperature. We are taking our protein 25 millimolar amyloid beta, we dissolve in some phosphate buffer in absence of ionic liquid and in presence of ionic liquid, right. And we acquire data of this 400 microliter of sample into 1 mm optical path, and we collect over 20 minutes to 40 minutes. We are measuring with say these are experimental parameter, bandwidth 1 mm, scanning speed 50 nm per minutes, and response time is 2 seconds. And we are like doing the analysis using one of the software called Bestel. Right. So, this is the result. Look at what happens to amyloid beta. Amyloid beta as such, right, as such, it is transitioning from a say random coil state here to a beta sheet state. So, blue to red, one can see it. So, what is happening here? If you just monitor at 217, the peak is going down, right. So, 217 beta sheet decreased over the amyloid beta formation due to aggregation, right. Now, you add this ionic liquid to this. Now, what is happening? You see the aggregation has not completely stopped, but look at the percentage of aggregates that has formed, quite drastically low. So, here also some probably beta sheet is forming, but that is low. So, if you just put it the aggregation kinetics, do the analysis, you can see in say about 400 minutes it completely forms the cross beta or beta sheet structure, whereas in presence of ionic liquid still not completely gone, right. So, here is something, and it is still 400 nanosecond, only 10 to 15% has formed. You can take another one, again same result, one can see it, it is a slowly 400 minutes going down, but here after coming here and then it plateaus down. So, it does not transition all the way from all the amyloid beta protein going to beta sheet structure. So, one can conclude that this EMI-MS ionic liquid inhibited the first step of aggregation, first step here. You just look at here after this, it's good. So, it inhibits the first step of aggregation in amyloid fibers, delays the aggregation, that is what one can see it. Now, one can really do the secondary structural analysis, how change are happening, and by deconvolution you can have alpha helix, beta sheet, random coil. So, helix, strand, turn and others. So, one can see now strands is going down and helix remains same, turn is there. And here also it basically amyloid beta in presence of EMI-MS, the beta sheet formation does not happen too much. So, result demonstrate that combination of CD and Bestel program can effectively evaluate the secondary structure of amyloid beta in details, right. Let us take another example, where we are tracking the amyloid beta transition from an random coil state to beta fibril state and show how an inhibitor can modulate this process, and that this is the way one can have the drug discovery for Alzheimer's, right. So, this is basically taken from the circular dichromosome spectroscopy of beta amyloid aggregation and the effect of inhibitors, right. So, we are focusing the amyloid aggregation, right, which is in the Alzheimer's disease. We are using CD to monitor secondary structure change that is happening and evaluating the impact of inhibitors, right. So, that is what we are doing, and key findings were in this paper, you can read it. The amyloid peptides start as a random coil, that is negative band at 200 nanometer. The other aggregation promoting condition, they transition into beta sheet rise fibril, that means we are going to get a negative peak at like something like this. We are starting with a positive peak here and ending up a negative peak at 217 in amyloid beta, and we are basically CD can display the capture of that structural transition that may have. So, that is what I was saying, if you have a CD like this 195 205 for random coil and then we transition into 217 for beta sheet, right. So, CD can detect like even the micellar intermediates or oligomers that are populated before you go to mature fibril formation, and these intermediates are important because they are neurotoxic. So, you know when aggregation happens, it starts with a monomer, forms the oligomers, that oligomers can be 2 mer, 5 mer, 10 mer, 100 mer, and then it goes to mature fibril. Research over here shows that these intermediates are very toxic because they can accelerate the aggregation kinetics. So, you can track their structural property and then one can understand the effect of inhibitors. There are several compounds has been tested which arrest this transition from random coil to beta sheet, and these attenuates the beta sheet signal, indicates that disruption in the fibril formation, and these compounds can stabilize into non-toxic conformation that can delay the aggregation, right. So, significance of this, CD basically a rapid and sensitive tool for monitoring the amyloid aggregation. It provides the screening method for say anti-amyloid drug, and it can complemented with another technique like thioplamine T. Thioplamine T is a dye that specifically bind to cross beta sheet structure of the amyloid fibers. So, you can combine CD with THT fluorescence assay, and if you want to visualize what kind of a structural change happening, you can use electron microscopy. So, these three techniques in tandem can track the structural transition that happens, and this highlights the importance of targeting early aggregation intermediates for therapeutics. The limitation is it gives you global secondary structural information. We do not have a atomic resolution details where actually this inhibitor binds, how they binds, those we cannot do. For doing that, we require probably to do NMR spectroscopy, where we can monitor each residue wise transition that may take place. And if you want to solve full structure, you can use the cryo-EM or solid state NMR for getting the full structural characterization. Right. Let us take another example. You know, there is another protein called amylin or HIAPP or human islet amyloid polypeptide. This amylin or HIAPP co-secreted with insulin from your pancreatic beta cell. Insulin, you know, helps in lowering the blood glucose level. Amylin basically promotes the satiety. So, if amylin is secreted into 1:100 ratio with insulin, that promotes the satiety. But in case of type 2 diabetes, this protein basically forms the amyloid. Right. So, IAPP amyloidogenesis happens in type 2 diabetes, and that amyloids start rupturing the insulin producing beta cell, and therefore, insulin production goes down. Right. So, like monitoring this or understanding how this protein aggregates and can we come up with an inhibitor for amylin or IAPP is great way to probably manage and treat type 2 diabetes. So, for progress of IAPP amyloid, it forms the it follows this sigmoid kinetics, which maps the structural transition. So, first transition lag phase, right. So, if you monitor this, this goes something like this: lag phase, elongation phase and saturated phase. So, here is a lag phase, right. This is elongation phase and then saturated phase. So, at starts of incubation, CD signal will show a negative minima at 202, because this is again an intrinsicular disorder protein. Then when protein starts segregating, CD detects transient alpha helix formation. So, I have a paper in biochemical journal when you can follow it. So, we have the typical alpha helix or helical formation. So, you see a 2 peaks that are populated 208 and 222, that shows that it goes through an alpha helical formation. And then growth phase, as aggregation proceeds, spectrum shift in real time, and then you have a negative peak at 200, which was from random coil, that disappears, and as we progress further, a beta sheet characteristic starts appearing at 216 to 218 and a positive peak at 195 to 200. So, that we know it is a characteristic of a beta sheet. So, from random coil going through a helical intermediate come to the beta sheet, that is what happens in amylin. And CD basically you monitor using CD. So, first is a random coil, then you have a helix and then you get a beta sheet. This kind of transition, random coil, helix and beta sheet, that kind of conformation you see. And now you have to combine probably that with a thioflavin T, because that monitors the aggregation kinetics just by probing the amyloid beta formation. So, you can use the Bestial algorithm again to extract and quantify beta strand fraction with time, and this allows to calculation of like rate kinetist, conformational conversion independent of like a mass aggregation with thioflavin T data. So, what one can conclude here, CD again versatile and indispensable biophysical tool that provides the chiral signature of molecular architecture by measuring the differential absorption of left-handed and right-handed circularly polarized light. It offers a limit window into folding, stability, interaction of most critical biological macromolecule. We can study the protein secondary structure fingerprint as we discussed. So, in protein science, CD is a primary method for global fold analysis of polypeptide, the sensitive to geometry of polypeptide bond backbone, and it provides a distinct spectra that allows quantification of a helix. These are the signature for helix, beta sheet or random coil. And we can use CD for understand the protein stability. We can actually calculate the Gibbs free energy that we had discussed earlier, right. So, not only this, I am going to talk to you in the next class how CD can be used for monitoring the DNA conformation and what kind of structural transition that one can have. So, just to conclude, the amyloid fibers that we are studying, the AIAPP or A beta, two of these protein we took. CD is uniquely suited for understanding the chameleon like nature of this amyloid forming protein. Chameleon like, why I am saying? It changes its structure, start with a random coil, this IAPP goes to helix, then goes to beta sheet. Now, this guy starts with a random coil, goes to beta sheet, but if we put micellar condition or the lipidic condition, this may populate helical conformation as well. So, it is studying the chameleon like structure of amyloid forming protein, and one can monitor this chameleon like property in a real time. It tracks the pathological transition from a disordered monomer to a toxic helical intermediate and finally to insoluble beta sheet fibers. And one can actually detect the oligomeric state using fluorescence dye that binds to basically large fibrils like thioflavin T dye, one can do it. So, CD can monitor the structural change that happens even though small and soluble oligomer, one can detect what kind of structure they populate. But as I mentioned, the limitation of CD, it cannot give you high resolution structure, it gives you global structural change that is happening. So, if you want to study the high resolution structure of any of such protein, you have to combine these with liquid state NMR and look at the structural transition happens. For the mature fiber that is formed, you can employ the solid state NMR or the cryo-EM to understand the structure that is populated. So, you know, biophysics is quite complementary, as we discussed that CD is not enough, you have to employ the thioflavin T fluorescence, the EMS structure, the AFM, the NMR, all of these can solve the problem. So, therefore, all these say complementary technique or orthogonal technique are required to delineate or deduce a conclusion that can come from this biophysical, biochemical problem that we are addressing in science. So, next class let us look at how we can use the CD for DNA structural transition. Thank you very much.